for the introduction, Bill.
In this presentation, I will be discussing how patient-derived models can be integrated into drug discovery. So I'll give a quick overview of the value of patient-derived models in drug discovery, looking specifically at the in vivo PDX with mouse clinical trials and humanized systems, in vitro 3D organoid drug screening and co-culturing, and then ex vivo patient tissue models. And then I'll give some examples of how these models have been applied for specific therapeutics and biomarker discovery. So although there are many reasons for the high attrition rates in oncology, the preclinical models come under scrutiny for failing to better predict efficacy. And therefore, this highlights the need for better translational models for both in vitro and in vivo. So patient-derived models are generated directly from the patient, and they may bridge that gap between the lab and the clinic much better than some of the traditional models because they preserve the original tumor histopathology and molecular pathology. They recapitulate intra and inter-patient heterogeneity, as well as patient diversity, and they're also pharmacologically predictive. But one of the challenges of these models is firstly accessing patients to generate the models, and then secondly accessing the models once they're established, especially those that represent patient heterogeneity and treatment history that is reflective of the current treatment landscape for specific cancers. So at Crown, we have developed platforms with patient-derived models, which include in vitro organoids, primary cells, in vivo PDX, and ex vivo patient tissue for different cancer types. And these models are integrated into our service platform to support drug development and translational research. So we have a range of capabilities from discovery to translational solutions, which is summarized here. And the patient-derived tissue, the organoids, and the PDX models can be integrated throughout the different stages of drug discovery. So you have models in our in vitro discovery solutions, in vivo pharmacology, and translational oncology. And these models can be standalone, or they can be integrated with other complementary services such as biomarkers, as well as in silico solutions. So a biomarker team work closely with you to design the study, to optimize it and maximize the preclinical studies for the specific objectives beyond just the efficacy readouts. Biomarker discovery can be incorporated early in studies like in vitro or screening with sufficient numbers of models, and then ensuring a statistical framework is appropriate to interpret the pharmacological data and then validate the biomarker and also provide insights into downstream studies or even the clinical trials. And to support the preclinical studies, we have a range of omics capabilities to dive deeper into the understanding of the target and the drug effects. The biobanked models are characterized genomically and via transcriptomics, and we also have authenticated each model via deep sequencing as an alternative to STR profiling. We also have 4D data independent acquisition proteomics and phosphoproteomics, plus a suite of spatial biology and digital pathology, immune profiling, as well as bioanalysis for DMPK within this core lab service offering. The baseline data generated through our omics platform, as well as the treatment data from our pharmacological studies for different cancer therapeutics is also shared within our databases, which are shown here. And some of you may already be familiar with our complementary online model databases. If not, you can register for access through the website. This is usually a very good starting point to help find suitable models. And there are 4 different databases, and I will be referring to Hubase, which has over 2,400 PDXs, and Organoid Base, which has over 600 organoids. So there's a lot of models, but the categorization to cancer type and can be found. There's filters that can be applied, or the search engine used to make it easier to find and explore the models. The characterization of the models includes genomics, proteomics, growth characteristics. There's model-specific information like mutations or resistance, pathology, and clinical information. The patient-derived models provide a wide range of opportunities for understanding drug response, as well as sophistication in the type of data that is generated. So here the in vitro, in vivo and ex vivo systems can be represented as either having higher throughput versus more complexity, which typically will have lower throughput. So in vitro, this will be primary cells from the patient tissue in 2D cultures versus 3D, such as organoids. And then there'll be more complex, such as co-cultures. And I'll give it some of these examples later. And also give examples of where there may be some matched models as well from in vitro to in vivo. And then similarly in vivo, you have the subcutaneous models versus orthotopic, and then more complex humanized systems. Ex vivo, you have multiple samples for bioanalysis or tissue for screening, or tissues slices for more complex profiling. And then similarly, the readouts, depending on the sort of assay, can be high throughput like tumor kill or genomics, transcriptomic analysis, or more complex such as proteomics or where specific imaging capabilities are applied. And then the in silico support as well can be more complex based on the data that's already available versus the data that's generated on study from the multi-omics approaches or the combination analysis. So the key takeaway here is the flexibility and also the throughput are really dependent on how the patient-derived models are used and the types of readouts. So let's start with PDX and how these can be integrated, and then I'll go back upstream to in vitro and then to ex vivo models. So the PDX collections have expanded in the last 15 years as a result of increased success of establishing and biobanking large collections, as well as characterizing each patient model. And the key differentiator here from other mouse model systems is the maintenance in vivo rather than the adaptation to plastic, and the preservation of genomic integrity, heterogeneity, and the ability to expand to apply indefinitely to different types of studies. These can also be used orthotopically to better understand the tumor microenvironment, can be used in human studies, and also to create organoids for matched models. And then PDX can be applied in different ways, and this can be early discovery, which is shown on the left-hand side, such as target identification, validation, for early drug screening in the form of organoids, mechanism of action, PK/PD, efficacy studies, which are dependent maybe on the particular type of therapeutic or target, and even the immunotherapeutic or the type of mechanisms such as resistance. And these can all be done in sort of smaller scale with selected models. The larger scale studies, such as mouse clinical trials for biomarker discovery, for patient stratification, indication expansion, all require a large number of models. And then you have the PDX development from new tissue direct from the clinic or the patient for a more personalized medicine or treatment approach, or co-clinical trials to inform directly on individual patient responses. So our aim with our collection is to represent as many different patients as possible with their unique features. And this gives a high-level overview of our PDX biobanks. Some of the key features, applications, the benefits, all represented by the different boxes. So for example, the breadth of collection is shown in the top row, and the different types of cancers in the pie chart. The full characterization that's undertaken in the second, and then you have the database on the right, which is HuBase. Our HuPrime collection is for solid tumors, and our leukemia models are for liquid tumors. We then have next the unique models within the collection, and some examples I'll show later as well, and also the integrated applications on the bottom row. So if we start with the unique collection, so the next generation of PDX models are those from patients who have undergone treatment to first, second, or third line treatment recently in the last sort of decade or so, and these are reflecting the new therapeutics that are being used to treat these patients. The response can be poor or patients relapse, and a sample is taken for generating the PDX. So these patients can have quite aggressive late-stage cancers that have relapsed from potentially multiple different types of therapeutic strategies, so they can actually be very unique. So the diagram here on the left shows the spread according to the cancer type, and bearing in mind this is an ongoing collection, so the number of models will continue to grow. So we have just recently added some AML PDX models from patients with prior treatment, and these models are either sub-Q or systemic models. And then on the right, this is a summary of some of those treatments within this collection. So there's still a lot of chemotherapy, as this is still the first-line treatment for many cancers, and also unspecified therapeutic or investigational drugs. So the information on these are not typically released. But these patients are enrolled to these trials as they've failed all other treatments. There's also combinations, targeted agents, which some of these include new agents like KRAS inhibitors or ADCs, and I'll share some examples here. And also you can see there are a number of models that have immune checkpoint inhibitors as well as immunotherapeutics that could potentially be quite interesting. And the database, you can actually filter for these models. But again, please reach out to us as there are some models that still may be in development. Another collection of unique models includes models that have specific resistant profiles. And these can be either intrinsic, acquired, or even induced or engineered. And at Crown, we actually have an extensive collection of resistant models, and there are several strategies or solutions for developing these models, which is broadly represented in this top section here. So firstly, and most ideal, is to acquire from the clinic, like the next generation of models that I've just showed, which we know is not always possible. So alternative approaches are the next best options, where the models can be created with drug challenge, either in vivo or in vitro. You can engineer them with CRISPR, for example, or you can use advanced technology to maybe create new models such as organoids or surgical models with imaging. And then the boxes here represent the different resistant phenotypes. And you can see a number of sort of PDX models here as such to targeted agents where resistance has emerged, or chemotherapy resistance. And these are more translational oncology models in comparison to the cell line models, which are typically used in early discovery. And you can learn more about our strategy for resistance in our on-demand webinar, and also I'll give you some examples as case studies. Other unique models include underrepresentative indications like prostate cancer, which actually are quite challenging to establish in vivo. So here is a summary of the prostate PDX models from patients who have received standard care treatment. A typical clinical landscape is also shown here with the different sort of treatment types and the options. This is based on the Gleason score and the staging, and then how tumors can relapse and the next treatment options based on hormone sensitivity. So metastatic castrate-resistant prostate cancer is an area of high unmet need and associated with poor survival, as the treatment options here are quite limited. So for the new PDX models, you can see here that the top three are derived from the primary site following radical prostatectomy, whereas the others are derived from metastatic sites, and all have had prior treatments such as hormonal treatment, chemo, or targeted agents such as enzalutamide, non-steroidal antiandrogens. Some of the models' characterization has identified TMPRSS ETS fusions and or PTEN loss, and also high levels of PSA expression. And here in this model, which is for metastatic castrate-resistant prostate cancer, no significant response is observed following treatment with either enzalutamide or abiraterone, but this was still shown or found to be sensitive to docetaxel. So these prostate models and their characterization means they have been, or they can be classed as either pre-treated PDX models, resistant models, but also as rare indications in that sort of in this sort of boxed categories. Next, I'm going to jump to the application of mouse clinical trials. And so this is a typical phase 2 like in vivo study to evaluate drug efficacy. And here the patient can join both arms as a PDX. There are 6 patients shown here with different colors, and these are recruited to both of those arms. And this the 6 patients represent patient heterogeneity, and they're treated with both the control or standard care in comparison to the test arm, which may also be a combination. So it acts as a self-control in this trial. There are 3 mice per group, per patient. So there's more replications in each arm, which captures intratumor heterogeneity, which is something that we see with PDX models. And the mice are recruited to the study when they reach a predefined criteria, such as a tumor size between 100 to 200 millimeters cubed, and the mice are then randomized across the two groups. So this is referred to either a 3+3 design or a 2x3 design. You can adapt the design to a format for your specific objectives. So, for example, you can increase or decrease the number of mice per group. So here you may want to use large numbers of models and then use a 1-by-1 format, which is sufficient and can be statistically powered. You can also focus on the type of cancer to discover biomarkers, or you can focus on a common target across different cancer types. And our white paper here actually can give further details and guidance on how to design and run a mouse clinical trial. So our sort of step-by-step guide on how to design and execute the mouse clinical trial has recommendations from multidisciplinary teams that support these trials in-house. So the bioinformatics team, for example, can advise upfront on the best practice and requirements based on the study design for the number of mice, arms, advise on statistical power. This is also being published in the BCM cancer paper shown here. And the scientific teams will obviously design the protocols, manage the studies, and also provide data in real time, which is shared through CrownLink, which is our customer portal. And then the biomarkers and bioinformatics teams will coordinate and translate the data into clinically relevant guidance. So that could be either patient stratification, biomarker identification. It could be just looking at the sort of different trial designs, also the sort of combination strategies that could be employed. So here is an example of how a hypothesis-free biomarker discovery could be generated without knowing too much about the drug target. So this is a real example of how we identified biomarkers in a mouse clinical trial of 77 PDX models across 4 different cancer types. And this is for a certain drug that was ready for phase 2 clinical trials. And we proposed 6 biomarker candidate sets, which are based on different sort of mutation and gene expression levels. So the first 4 were mutation-based, whereas the last 2 were gene expression-based. So for instance, the biomarker candidate set 2 is a mutational biomarker for current enrollment into the clinical trial. The class 4 is a newly added enrollment criteria for which was based on our findings. And the class 1, which was again also a mutational biomarker set, but with multiple genes, had a better separation for responder and non-responders with higher resolution, but this wasn't selected, which could be due to the single gene biomarker being more easily tested in practice than a composite set. So this highlights the balance between predictive power and practicals for clinical use. So obviously there are limitations to the immunodeficient models, as the testing of human-specific immunotherapies requires a human immune response. And here we have some relevant publications that can advise on which models to use. PDXs can be used to evaluate cellular therapies such as CAR-Ts, which is shown here in the white paper and the publication. Or PDX can be used in humanized studies, which can also be more complex and costly due to the engraftment of human immune cells on immune deficient backgrounds. And this enables then the reconstitution of the immune system. And I'll share a case study on this later. But next, I wanted to move on to in vitro as an alternative or upstream tool to help refine some of the more complex in vivo studies. We have in the past used PDX tissue ex vivo in assays to provide a higher throughput screen before moving into in vivo studies. But the organoid technology actually has a huge number of advantages as it's maintained in vitro and not in vivo. And so it is pretty much a in vitro version of PDXs. And this also supports the 3 Rs. So for the organoid technology, so this is just an overview of our organoid biobank. We have an exclusive license to use the hub technology for creating and using organoids for preclinical oncology testing. So we have PDOs that are created directly from patients, and these are models that Hub have supplied, which is shown in the top panel. So these are referred to as PDOs. We also can create, using the same technology from our PDX biobank, the fully functional organoids from the PDX, which are referred to as PDXOs. And this is because the adult stem cells reside in those PDXs, just like they do in patients, and these are used to harness to create the organoids. So this means our vast PDX biobank is a continuous pool of patient tissue, and it makes it easier to create those organoids. And then we'll have downstream matched PDX models ready for pharmacological studies and translational studies. Details of the collection that was created, we've also published on this. It describes the model expansion and how we've created a working bank and a working process so that models are ready for service, similar to what we do with our PDX collection. And then also similar to our PDX biobank, our organoid biobank is also fully annotated for clinical information, pathology, diagnosis, genomics, and this is accessible via organoid base, and this can help with model selection. And this slide gives you a breakdown of the different cancer types. We have over 15 different types in our collection for our PDXO collection. As we have thousands of PDX, we're growing this PDXO collection continuously, and the database is updated in parallel. The PDO models on the right include both tumor and normal tissue organoids. And again, that's because the adult stem cells resides in all tissue, including normal and disease. So we have over 6 different indications, as well as, in some cases, matched metastatic and primary tumor pairs. The working biobank actually makes it more efficient to integrate organoids into efficacy testing in vitro across large patient populations, molecular subtypes, to really kind of identify responder and non-responders, also look at resistance, and also help refine downstream in vivo efficacy studies. And this can be done with a broad range of different therapeutics, from small to large molecules, novel agents like ADCs, and also oncolytic viruses. We can investigate the combinations. We can look at predicted biomarkers, off-tumor effects with the normal organoids. And you can find more about the applications in our white paper, which is shown here in the middle. And also there's a review paper in current protocols on the relevance of organoids and the matched models, and when to select then in comparison to other 3D systems and PDXs. We also reported on how the in vitro response compares to the downstream matched PDX model. So in this slide, it summarizes the data from 30 matched data sets, just again to demonstrate the correlation of response profiles between the organoid cultures and its associated in vivo PDX model. So the IC50 is on the x-axis, and anything less than 1 micromolar is sensitive. And the TGA, which is on the y-axis, anything above 70% is defined as sensitive. So this Pearson's correlation plot can be divided into different quadrants, where sensitive models in both systems fall in that top left quadrant, and the insensitive fall in the bottom right. And this has also been tabulated to represent the 4 different corners. So for PDXOs, there's a trend overall for the positive prediction where the organoids data showed sensitive similar to the PDX models, and this was approximately 75%. There's also negative prediction with the organoid predicted insensitive response, which was about 91%. And overall, this shows a prediction of 86 out of the 30 models, which is across 4 cancer types, 11 different models, and 12 different test agents. So this is quite encouraging. And obviously, there are some therapeutics where it won't predict, such as immunotherapeutics, due to the lack of the immune cells. We found cetuximab wasn't predictable, and even some targeted agents. So sorafenib, for example, the cutoff for the response was outside of the dynamic range. So it didn't correlate according to this criteria, but this is just generally for guidance to give an overall demonstration of the biological equivalence of the correlation, and generally favorable for most of the agents that were included. But obviously, some adjustments can be made according to specific therapeutics. And then generally the 3.8-well format allows high degree of flexibility when working with patient-derived models, such as the organoids, but also the PDX spheroid models, seeing as some PDX models, we don't have matched organoids, so we do have to still rely on using the PDX as a source of tissue for some of the in vitro 3D spheroid modeling. And there are a number of different options in the setup to achieve different objectives. So you can identify a lead compound. You may want to opt for a library screen. You could look at the full dose profile efficacy, determine which compounds are most efficacious. So you could look for that in an efficacy screen, or you could look at a combination screen to evaluate multiple strategies before moving into an in vivo situation. And this can be done again for a range of different therapeutic compounds, such as small molecules, antibodies, ADCs, peptide conjugates, oncolytic viruses. And then the endpoint readout typically for these assays is a CTG, but other types of viability can also be used. And then for the more complex biology, we apply high content imaging, which gives more insight into the efficacy and mechanism of action beyond just that tumor kill. As well as the automated screening service, we also have an automated biomarker discovery platform to analyze large data sets generated, very similar to our PDX in vivo. So the high content imaging allows multiplexing to monitor multiple features within that microenvironment, and this can all be done in one well. This slide just outlines that typical sort of study process, so you can see how easily the design can be integrated. And then you have that added value that you can measure hundreds of different phenotypic morphological parameters simultaneously. Some of them are listed here, which you can do without the need to repeating the experiment or using multiple wells. So this is including the number and the size of the cells, the tumoroids, looking at apoptosis, invasion, migration, immune cell infiltration, tumor killing. So quite a broad range of phenotypes of interest. You can also measure tumor killing in both tumor and normal organoids from the the PDOs. And the high content is readily scalable for high content screening and can be quantified by image analysis using proprietary software, which measures distinct phenotypic and therapeutic responses beyond just those that tumor kill. And to make the organoid screening more accessible and efficient, we have set up Organoid Explore, which is an organoid panel screening to provide drug response profiles in tumor organoids and also a handful of normal organoids. This runs every 3 months in pre-selected models in as little as 6 weeks. It's a 5-day incubation with CTG readouts. So it's quite simple. You get IC50 curves or drug combination analysis that can be coupled with downstream biomarker analysis. The panel consists of both PDOs and PDXOs, and that's across 7 different indications. But you can actually screen compounds in either the full set or in subsets based on the sort of mutation profile, indication, or a specific biomarker. So, for example, the KRAS panel across 4 indications could be an option. And then there's also an ultra flexible panel where you can select 20+ models to meet specific research needs. Now, similar to the PDX model, inclusion of immune cell components, it's critical. It's also critical for evaluating immunotherapeutics in organoids. The tumor microenvironment is therefore reconstituted with the inclusion of immune cells, but this may also, just like the in vivo system, may result in allergenic reaction. Alternatively, you've got the native tumor microenvironment in ex vivo patient tissue, which provides an alternative option when investigating immunotherapeutics, which specifically target T cells. And the complexity of the tumor microenvironment is still intact. So there is autologous immune cells and stromal cells, but there is a limitation here of the scalability and access to that patient tissue. So we did actually have a series of tech talks and podcasts, this ebook, which has protocols, and this all dives into advanced in vitro technologies like organoids, high content imaging, tissue analysis, and how these are impacting immuno-oncology drug discovery. So you can access this again online. So there is a lot of flexibility with Immuno-Oncology 3D co-culture assays. And this is just kind of depicted here to show the differences with some of the organoid setup. So both organoid and spheroids can be used with different immune cells. And this can be used for different types of therapeutic agents. And these are co-cultured to evaluate the response to immune checkpoint inhibitors. You've got cellular therapies, immune cell activators. We are also doing triple co-cultures which include cancer-associated fibroblasts within the organoids and the immune cells. And this mini tumor microenvironment is currently being evaluated and tested. We then apply high-content imaging, which provides specific advantage here to enable multi-cell readouts effectively and efficiently. So, for example, you can look at the organoid measurement specifically, the immune cells as well. You can look at the immune cell positioning, the sort of trafficking and the migration and the infiltration, and correlate that back to the response. The ex vivo patient tissue platform is potentially the most patient relevant and complex, as it utilizes fresh patient tissue which is processed within 24 hours. So this really does preserve the native tumor microenvironment, those endogenous immune cells and stromal cells, and as well as the heterogeneity of the patient. And each patient really is very unique, and that's reflected in this TME. The assay is quick, as there's no culture or establishment in vitro. This is a 384 well assay, so multiple compounds can be tested across multiple patients. Drugs such as chemotherapeutics or standard of care agents and immunotherapeutics are assayed over 5 to 7 days before being fixed and imaged. The phenotypic readouts go beyond, again, that standard cell viability when high content imaging is applied. You can also do cytokine analysis, immunofluorescence, and where possible, the sample from the clinic is characterized. So we understand exactly what maybe the immune niche composition is of that tissue. And this can be quantified by IHC or by FACS to understand the makeup of that tissue, and that then can be correlated back to the response. If there is sufficient material, the tissue is also banked or organoid are generated. And you can, again, find out further information through online webinars. We have optimized the platform for a number of different indications, as shown here, as well as custom offerings for other cancer types. Now, tissue sourcing for specific subtypes or pre-treated patients can be actually quite limiting, but we can advise on the best options and timelines. Okay, so in the next part of the presentation, I'm going to give you some specific examples of how the patient-derived systems have been applied to drug discovery projects. There's quite a number of different types of modalities that we've tested, but really there isn't time to cover all of these, so I'm just going to pick up some highlights. And we would be more than happy to discuss agents more specifically, such as ADCs or cellular therapies or other targeted agents. So please feel free to reach out to us. So we start with immune checkpoint inhibitors, and the first case study is assessing the immune checkpoint inhibitor response, such as pembrolizumab and ipilimumab, using the ex vivo tissue that I just described from a range of different cancer types, which includes non-small cell lung cancer. So the response is graded as sensitive or resistance according to the positive immune stimulator agents such as the superantigens, and this is to compare the immune-mediated response in the study here. And the tumor volume is then quantified and measured by high-content imaging. So in non-small cell lung cancer, you can see that there is a mix of response to pembro and ipilimumab, but generally there is a 20% responsive population, which aligns with the clinical data for pembrolizumab. Now, you can also look at individual models more closely to understand why they may be more resistant. So, for example, in this particular sample, which showed a strong decrease in tumor size and immune cell count upon treatment with SEA, there was only a mild response to pembrolizumab. So further investigation into the phenotypic response was conducted with cytokine analysis, and you can see an increase in interferon gamma and granzyme B, interleukin-2 upon immune stimulating treatments, which supported the immune-mediated killing by SEA. But this obviously wasn't seen for pembrolizumab. So the baseline monotherapy data can then be used to determine in combination testing to try and enhance the immune checkpoint inhibitor killing response. And this could be dependent on how hot or cold the tumor is and the level of immune cell infiltration at the time of the tissue that was collected. And this can be determined by tumor histology or for immune cells or cytokine analysis. And here in this example, a number of different immune checkpoint inhibitors, either targeting PD-1 or PD-L1, were benchmarked against different combinations, which may increase the combination may increase immunogenic cell death. So that could be with targeted agents or inhibitors for CSF1R, which targets a suppressive tumor microenvironment, which is mediated by the myeloid compartment with tumor-associated macrophages. And these types of studies can be quite informative because of that autologous and intact tumor microenvironment. However, the in vivo studies may still be required before moving into the clinical stages to determine efficacy and toxicity. So those types of studies could look like something like this. So in this second case study, Keytruda was tested in a panel of small cell lung cancer to identify patient responders. And this is utilizing the CD34 reconstituted humanized mouse model. And this really allows us to leverage the larger collection of PDX models to run those humanized clinical trial studies, although this is quite complex due to the donor-donor variability, reconstitution, and the potential alloreactive responses. So some pre-screening is advisable for the models with the donors, just so that the alloreactive response is manageable. So in this design, 5 donors for CD34 stem cells were used to construct the humanized mice, and 30 small cell lung cancer PDX models were screened for PD-L1 expression by FACS. And then 7 were selected and moved into the pharmacological studies. So 1 tumor was implanted per humanized mice. So you can get a checkerboard of 5 different donors across 7 different models. And this was in a 1-by-1 design. You can see the variation across the checkerboard, and this is shown nicely, summarized nicely in the waterfall plot. And here you can see that there is a 36% response rate in the small cell lung cancer PDX, which actually did correlate with the clinical response to Keytruda. Further investigations into the infiltrating T cells showed an increase in the percentage of T cells in the tumor from the responder PDX, whereas there was an increase of infiltrating macrophages in the tumor microenvironment for the non-responders. On the right, it shows a relative expression of the CD38 positive M1 macrophages polarizing in the responders in the top diagram, and then the M2s in the non-responders. So the humanized mouse clinical trial actually provided great insights into the potential mechanism of response or resistance and the potential for biomarker response. But one of the major reasons for failure in the clinical is that drug resistance, and the mechanisms of resistance can be multifaceted. Thus, having relevant models to tackle specific resistance phenotype is essential for, especially for those next generation of agents or combination strategies. So, for example, new agents like KRAS inhibitors, the mechanism of resistance is emerging in the clinic, and new models are needed to reflect this new resistant phenotype. So we have several approaches as described in addressing resistance, but I'm just going to focus on the intrinsic or acquired resistance that can be evaluated in organoids or PDX models. So this next case study highlights how Organoid Explore, the full panel, can be used. It shows differential efficacy response for targeted therapy agents with different mutational backgrounds. So the KRAS mutations are quite diverse in the panel, as shown in the heat map, and the AMG 510 response can be seen in the G12C models, which is expected, although there is still a variation in that response as well. So the organoids provide a fast and effective way of profiling many different organoids, patients, and potentially identifying those low responders or non-responders in the models, and also looking and correlating this against secondary KRAS mutations such as G12D or Y96C. And also this can be used for potentially for combinations with other targeted agents such as SHP2. The models can be selected for evaluation for in vivo studies, and we actually have in vivo models matching these organoids. So here, this is a lung PDX models, which shows significant TGI response in this model here. But there's not a complete response. There's still some residual tumor growth. So it's not complete tumor regression. And this can then be used to generate or induce a model resistance through sequential rounds of drug exposure, which is what we did here, and then capture those outgrowth tumors and reimplant them to eventually we see no response and resistance to KRAS inhibitors. So this model is still undergoing characterization so we can fully understand that mechanism of resistance. We also have pre-treated patient PDX models which have KRAS mutations and different sort of treatments that they've undergone. So this model summarizes the colorectal cancer models. And here patients are receiving first-line treatments to FOLFOX, second-line, third-line to different agents, and then also in some cases KRAS G12C inhibitors such as Krazati or AMG, sotorasib, which were only recently approved for a non-small cell lung cancer. Now, in this particular patient that's just highlighted, so these are 3 models that are derived from the same patient. So this was a Caucasian female who had metastatic adenocarcinoma with a G12C mutation. Initially, there was very good response to Krazati, which was given as an investigational treatment in combination with SHP2 inhibitor. So a sample was created as a PDX. The patient soon relapsed with biopsies from the liver were then taken, and this created the second model. And then eventually the patient went on to a fourth investigational drug. So these 3 models are longitudinal models for KRAS inhibitor sensitivity and resistance and are being characterized in vivo currently. So here you can see the growth profiles of those 3 PDX models, which are subcutaneous from that same patient at different stages of disease progression and treatment relapse. So the original model shown here from the patient who, when they were initially responsive to KRAS inhibitors, shows good TGI in PDX. But again, there's not complete tumor regression. And then in the relapsed models, so 9537 and 9547, there is weak response to Krazati, similar to the patient, and also a sort of a partial response to AMG 510. But this is still representing progressive disease, or still referred to as resistance. So these serial models of specific oncogenic mutations and response to targeted agents and then relapse are extremely valuable for understanding the disease resistance. And these are undergoing, again, for further characterization and understanding of what that mechanism resistance is. And we do actually have other KRAS inhibitor resistant models that are in development, as well as organoids that are being created so that we can add them to our KRAS panel for in vitro screening. Now, in addition to the example provided earlier about mouse clinical trials for hypothesis-free biomarker discovery. I just wanted to kind of mention another PDX mouse clinical trial, specifically looking at cetuximab biomarkers of response, and also an example for organoid screen and how that also can provide insights for biomarker identification. So here, cetuximab is used to treat patients with EGFR expressing and KRAS wild type metastatic colorectal cancer. But it has no effect in mutated cancers. So this is so there is actually a real-time PCR companion diagnostic test for KRAS wild type, which allows patient selection. There's also a diagnostic EGFR IHC assay, which can be used to detect EGFR expression, but it doesn't actually predict the response to cetuximab. So the objective here was to evaluate a predictive biomarker for cetuximab, and this was done in gastric cancer using a mouse clinical trial. So this study was also published in scientific reports using 20 gastric cancer prostate PDX models that were recruited in a 5x5 design to allow sufficient powering and also enable sampling for IHC. And we identified and clustered the therapeutic response and non-responders and found that actually EGFR expression, which is shown here for IHC with the copy number, with mRNA expression, correlated with the high response to cetuximab in 4 of these models that are shown here. However, the tumor growth inhibition response didn't really correlate very well with the EGFR expression. So, this led us to investigate alternative correlation. And so rather than using the sort of traditional tumor growth inhibition or area under the curve, we applied a more sophisticated linear mixed model method of data analysis, which examines the sort of heterogeneity within the patient and across patients. This was then able to provide a clearer picture for biomarker selection and actually confirmed that EGFR expression was the top ranking gene. And these findings agreed with the published phase 2 trial with cetuximab. And here you can see 7 patients, but all 7 patients in that high EGFR expressing category out of all of the 44 gastric cancer patients showed a reduction in tumor volume when compared to 37 of the low expressing patients with low EGFR. Only 24 showed a reduction. In a further phase 3 randomized trial, which was open label trial for advanced gastric cancers, again looking at cetuximab, it was concluded actually there was no benefit for the addition of cetuximab. However, when the results were reinterpreted for patients with just high EGFR protein expression, so this is above 200 in scoring, in IHC scoring, this actually revealed that cetuximab did provide clinical benefit for those gastric cancer patients with high EGFR expression. So the mouse clinical trial that was conducted did actually inform quite nicely, which correlated with the clinical response of alternatives for identifying the right patients for that treatment. We also had a poster at AACR where we leveraged the full panel of 50 organoid models to screen for sensitivity to several standard care compounds such as paclitaxel, and then through biomarker analysis, similar to what we do with the PDX, we could identify a gene signature of 28 genes that allowed us to accurately predict the response in that 50 organoid panel. You can actually download this poster using our QR code and visiting our website, or you could hear more about it in our on-demand webinar. What I want to do was actually just finish with a final case study focusing on lead identification using organoids for bispecific antibodies. And this study was published in Nature Cancer last year, where we were able to demonstrate the value of high-content imaging with organoids for unbiased functional testing, and this led to a lead candidate for clinical trials. So several organoid models were first screened with over 500 therapeutic antibody candidates to narrow down the selection to only a handful, and then these were screened in 30 colorectal cancer organoid models to see so this is a larger cohort of patients to select in the end a compound that showed the highest sort of selectivity towards the tumor organoids in comparison to normal organoid counterparts. and also in comparison to cetuximab. The data here shows the internalization and the degradation of EGFR by test agent MCLA-158, which was more rapid in comparison to cetuximab, and also mediated by LGR5, but not seen in normal tissue, which lacks LGR5. So this led to the identification of a single agent targeting EGFR and also LGR5 positive colorectal cancers with limited toxicity in the normal tissue. And this was also further validated in vivo efficacy studies where the colorectal cancer PDX model and IHC proliferation index scores also demonstrated the significance of response in vivo. And then this just summarizes the path to the clinic and the current phase 3 that is scheduled, and the sort of contribution of the patient-derived systems in the preclinical phases of discovery of this lead compound. And then just to summarize, so the patient-derived systems improve predictability, they preserve the original tumor histopathology and molecular pathology, heterogeneity and diversity, and also are highly predictive pharmacologically. The access to the patient-derived models is pivotal for drug discovery, and our large and diverse collections enables the patient-derived systems to be seamlessly integrated into drug discovery workflow, and our databases enables a selection of appropriate models to interrogate a range of different therapeutic modalities from biospecifics, immune checkpoint inhibitors, cellular therapies, small molecules, ADCs, and this can be used to determine efficacy, mechanism of action, resistance, patient stratification, and biomarker discovery. And then integrating those advanced technologies can provide deeper insights and potentially increase the likelihood of success. With that, I'd like to thank you for your attention, and I'm happy to take any questions.